Good morning, everyone. We'll just wait a minute here as we let a few more people trickle in before we get started. But thanks for joining us all.
Hi, everyone. While we wait, I'd like to cover some quick housekeeping items for today. At the bottom of your screen, you will see a chat box where we would love for you to say hello, introduce yourself, tell us where you're joining us from today. And right next to the chat box, you will see a Q&A box. Please type in your questions here so that it's easily visible to us and we can address them easily during the Q&A session. And you also have the option to upvote questions you'd like to be answered the most, so we can prioritize topics that are most relevant to the audience today. And yeah, we'll just wait another minute before kicking off. All right, I think we can get going. It's a good number in. All right, it's an absolute pleasure to welcome you all here to our webinar from a very gloomy morning here in Singapore. But we're really happy to share with you on our cell line development and utilization trends in the cultivated meat industry report. I'm Manasa, the SciTech specialist for cultivated meat here in GFI APAC, and I'm joined here by my colleague, Dr. Dean Powell, our senior SciTech analyst. Our webinar today will run for a total of 1 hour. Dean and I will begin by giving you a short presentation for about 15 minutes, where we will cover the key findings of our report. And after this, we will be joined by our lovely guests, who you can see here, Dr. Olivia Ogilvie, CEO and co-founder of OppoBio, and Dr. Gurugirija Ratnasamy, senior scientist at TurtleTree, for a 20-minute panel discussion to delve deeper into some of the concepts and topics that we've covered in our report. And finally, we will dedicate the remaining time for Q&A, where we'd love to address questions sent in from you guys in the audience in the Q&A box. With that, maybe we can start sharing our slides. To the next one. Awesome. For those of you who are not very familiar with who we are and what we do at the Good Food Institute, or GFI, we are a global network of nonprofit think tanks focused on building an ecosystem for alternative proteins to thrive. We believe that alternative proteins are one of the most compelling, scalable, and market-driven solutions for future-proofing our global protein supply. We currently operate as 6 affiliates with more than 150 staff around the world. And our vision is to make alternative proteins no longer alternative. And to do this, we work across 3 programmatic pillars to mobilize open access research funding, support foundational science and knowledge generation, and provide resources and support to companies, startups, investors, policy actors, and even governments to accelerate the sector. And as you can see, as a nonprofit, we are fully funded by philanthropy. Next slide, please. If we look at the cultivated meat industry, it has expanded rapidly over the recent years. And our internal numbers show that there were over 150 companies in this sector globally at the end of 2022. However, the availability and development of commercially viable cell lines is a bottleneck that is hampering progress. Next, please. To better understand factors underpinning this bottleneck and identify actionable opportunities to address them, we surveyed a pool of companies at the end of 2022. And these companies ranged from startups to more established entities that are either working towards developing cultivated meat products or working as enablers in this space by supplying the industry with cell lines, media, and other process inputs. And all of that effort led to us being able to distill a snapshot of this emerging industry's needs with an eye towards future cell line demands, characterization requirements, and regulatory considerations brought to you in this report here that you can see on the right that we've developed in partnership with BTI at A-Star. So diving straight into the data, we saw that one of the main challenges that companies are facing is sourcing cells from species deemed as high priority. Most companies, about 63%, are interested in obtaining cell lines from both terrestrial and aquatic species, as you can see in figures 2 and 3. For terrestrial species in figure 2, cow, pig, and sheep were the top-ranking species that companies said they were facing difficulties in sourcing cells from. Similarly, for aquatic species, salmon, tuna, and other finfish emerged as the top 3, with shrimp following closely behind. The fact that we see some species being preferred to others is indicative of a duplication of efforts in cell line derivation and perhaps adaptation and even culture optimization. Next, please. Yep. We also saw that there's a heavy reliance on slaughtered animals as cell sources. As you can see in figure 4 in the bottom right, close to half of the companies, about 48%, currently use cells sampled solely from slaughtered animals, and about 24% use a mix of live and slaughtered sources. However, only about 27% depend solely on live animal sources. And we saw that the 3 main barriers to accessibility to cell lines were access to animals, proximity to slaughter and cell isolation facilities, and the company's staff not being located in the right geography. Nonetheless, it was encouraging to see that companies are keen to move away from slaughtered animal sources, and 76% of them are keen to arrange contracts with farmers, ranchers, and even fisheries in the local region to address this sourcing issue. Next slide, please. We also went on to probe into what cell types companies are currently using and what is most desired by them. We found that the availability, commercial relevance, and the ease of culturing cell types have likely not shifted since a previous survey we conducted at GFI in 2020. As you can see in figure 5, companies reported the use of a wide variety of cell types with myoblasts, fibroblasts, and mesenchymal stem cells, or MSCs, as the most commonly used. Next, please. When asked which of these cell types were the most difficult to procure, it was clear that embryonic stem cells, or ESCs, and induced pluripotent stem cells, or iPSCs, were among the most difficult to source, regardless of whether it was terrestrial or aquatic species. As you can see in figure 8, it's important to note that myoblasts ranked the highest for aquatic species, even more than ESCs and iPSCs. And this may be driven by technical hurdles, such as an absence of validated isolation and culture protocols, perhaps a lack of technical expertise to do so, and inadequate molecular characterization tools, such as primers and antibodies for aquatic species that are less studied to date. Given the challenges with sourcing desired animal species and obtaining the right cell types, we started thinking, how can we think towards improving these aspects and strengthening cell line development in the industry to improve our chances of generating commercially viable cell lines? Our data shows that companies are largely pursuing cell line development in-house, which is a time-consuming and resource-intensive process. However, it was really encouraging to see that close to half of the companies are open to off-the-shelf options from commercial providers or repositories and government-sponsored cell banks. Next, please. To understand this better, we asked companies what they would anticipate paying for a vial of 1 million cells and what they saw as fair market price. Close to 60% of the companies, you can see the breakdown in figure 9, anticipate paying between $250 to $1,000 for a vial of cells. and the other 40% deem greater than $1,000 as an anticipated price that they're willing to pay. While more information is needed to delineate whether these prices are for cell lines intended for R&D use alone or also for commercial use, it shows a willingness of these companies to explore external solutions to boost the quality of cell line development for their products. Furthermore, our data showed that nearly 50% of the companies are also actively seeking cell banking services, which is important for storage and traceability from a regulatory perspective. This presents a golden opportunity for more industry enablers to enter the space, such as B2B entities, either alone or in partnership with universities, perhaps, to help fill in some of these gaps. For example, partnering with smallholder farmers, ranchers, and fisheries and in strategic locations to source cells from desired species, develop and sell cell lines of the most highly desired cell types, and even offering banking services would be of great value. So any of these solutions, either alone or in combination, would be hugely beneficial moving forward. So if there's anyone in the audience here who's thinking along these lines, please feel free to reach out to us, and we'd love to talk to you more about this and share our insights and see what we can do to help you in exploring some of these solutions. And with that, I'd like to hand over to Dean for the rest of the presentation.
Great, thanks for that. So now I'll look at a few of the other topics that we explored and saw as the key outcomes. So first was exploring companies' attitudes to genetic engineering. So we observed that there was really lukewarm interest in genetic engineering, with about 20% of respondents comfortable with genetic engineering for commercial purposes, while slightly more were okay with that or for R&D purposes. And when we asked this question, we specifically classified it as, would you use genetic engineering that would be classified as such for regulatory standard purposes in their target market? So this is really a question of genetic engineering when it comes to regulation. However, a majority of companies expressed a keen interest in using immortalized cell lines. And as you would expect from the previous data, that was mostly a preference for spontaneously immortalized cell lines, which from all jurisdictions, as far as I'm aware, wouldn't be considered genetically modified from a regulatory perspective, with lower interest in other methods, which will vary depending on markets. It is important to highlight that this survey did not explicitly poll interest for the use of gene editing, such as CRISPR-Cas9, for example, to make precise changes in a genome. And this is important as this is a separate method you could use to generate immortalized cell lines, where in countries such as the US and Japan, this isn't necessarily classified as a genetic modification process. And more countries such as the United Kingdom are actually leaning in this direction as well. So we may see a global increase over the coming years in the use of gene editing as a tool in the cultivated meat production process, if it becomes clear that this will not fall under existing gene modification regulatory guidelines. And so we see this as an opportunity for regulators across different regions to potentially align their regulatory guidelines related to cell lines developed via gene editing, which could allow for streamlined assessment of these processes and build confidence in companies employing them as a technology. So then if we look specifically at cell line adaptations and different types of functionality that companies can pursue within their cell lines, we actually asked respondents to rank up to 4 of their most desired cell line characteristics, as you can see in the table here. So as you might expect, the high proliferation rate was the most universally desired characteristic. And we also asked what kind of doubling time companies believed would be achievable for commercial purposes. So we saw about 80% believe that less than 24 hours doubling time would be achievable and is something they would desire. We then also asked a bit more on the topic of immortalization. So interestingly, you can see about 50% of companies deemed immortalization as a highly desired characteristic, which may seem kind of counterintuitive when you think about it, as cell line immortalization is often talked about as being quite important. But we then asked about what number of population doublings companies thought would be relevant for commercial purposes. And we actually found there was no clear consensus here on what amount of doubling would be required. So around 29% of respondents actually suggested a population doubling limit below 40 would be acceptable. And so between 40 to 60 population doublings is what's generally believed to be the Hayflick limit. So that's the amount of population doublings and non-immortalized cells would generally be able to achieve. We also looked at developments around serum-free growth adaptation. So of respondents, 64% had already achieved serum-free growth for at least one of their cell lines being developed. And most of this serum-free adaptation was achieved in 6 months or less. We then also looked at non-adherent cell growth as one of the other highly desired cell characteristics that companies are looking at. But only about 46% had achieved non-adherent cell growth so far internally. But those companies that had achieved it actually found it easier to do so while maintaining or improving the performance of their cells as compared to cell lines adapted to serum-free growth, where drops in performance were more noticeable and common. And so we see that this data in particular could really help inform B2B cell line providers of the characteristics they could preferentially target to make their products more attractive to the cultivated meat companies. And if we have greater availability of these commercially available cell lines with desirable functional and phenotypic properties, this could circumvent or reduce the need for all the cultivated meat companies and relevant academic research labs to invest additional resources in their own cell line development, as we're seeing at the moment. We also investigated companies' regulatory priorities and knowledge levels when it comes to regulation of cell lines. And this clearly reflected interest in the two markets that are leading regulatory developments, being Singapore and the US. But after that, there was widely dispersed interest across the globe. And so out of 35 respondents, we actually saw that only 10 companies, or about 30%, expressed confidence in their understanding of documentation and testing requirements for obtaining regulatory approval, specifically when it came to cell lines in their high priority markets. And we found the majority of those that weren't confident in their ability were just due to lacking internal information, whereas at about 26%, it was actually due to unavailability or inaccessibility of relevant info. And only 6% had yet to investigate regulatory requirements. And so some of the specific information gaps they highlighted were first, the assessment of techniques used to establish cell lines, such as immortalization or iPSC reprogramming, and how those would be regulated, as we discussed earlier, in relation to genetic modification. And the other area that was most highly seen as a gap was the testing and characterization requirements of cell lines, as opposed to source animal and cell line isolation and storage documentation, where companies generally expressed confidence with what information would be required from a regulatory perspective. So the twofold takeaway from this data is we see there's still a large job for governments and regulatory professionals to understand and communicate the requirements for cultivated meat, cell line kind of documentation and methods that can be used in different markets. GFI does maintain a list of technical consultants, including some regulatory experts, but this is quite small and we see a need for more. We can also see this as an opportunity for cell line providers to understand relevant regulations and tailor their products to be regulatory ready. This would be another incentive for customers to work with them. Finally, as a caveat, this survey was conducted just before the first FDA no questions letter, so understanding of US requirements has likely improved since this survey was conducted. And as a final point, we investigated religious certification and other documentation preferences. So a majority of respondents want to comply with halal and kosher guidelines, but only a minority confidently understood what the requirements for these certifications would be, about 18 and 13% respectively. From our current discussions and what we've seen from other groups, many Islamic religious authorities may not approve halal certification for cells taken from an animal prior to slaughter through appropriate methods. So this could become a major issue given that, as Manasa indicated earlier, that 77% of surveyed companies' future cell line development plans include incorporating cells obtained from live animals or both live and slaughtered animals, which would not necessarily fit with their desire to also be able to be halal certified. And so just to highlight a few of the key takeaways from this project, as we've both said now, there's almost certainly significant duplication of effort with similar species, cell types, and product characteristics being pursued by many companies, and cell line development largely being conducted through resource-intensive in-house efforts. So we see this as a big opportunity for B2B cell line providers to offer cell lines that tick off these common requirements and reduce redundant R&D. So companies did express an explicit preference for off-the-shelf options in this regard, as opposed to engaging in ongoing research collaborations with a cell line provider. And this would also lower the barrier to entry for startups that would prefer not to develop cell lines in-house or do not have the relevant expertise. For example, if they're looking at developing culture media, scaffolding, or other bioprocess innovation. Again, we saw that strong demand for live source cells. However, there is that outstanding issue around religious certification and companies' understanding of this from a regulatory perspective. Again, genetic engineering for mortalization or other performance benefits appears underexplored due to regulatory concerns. This is despite the potential for significant advantages that this could offer to the sector. This survey did not go into detail to understand active use of gene editing that could avoid this GM regulation. And this is an area that is worth exploring more and monitoring in an ongoing basis as regulatory regimes change around the world. And finally, that clear need for experts with appropriate technical and regulatory knowledge to assist companies in navigating approvals in different markets, we see as being really crucial, especially if these requirements are not aligned as more countries set up their regulatory regimes and begin approving their first products. And so with that, I'd just like to also say thanks to some of our other co-authors in GFI, Elliot and Claire from GFI US, as well as our partners at BTI A-Star, specifically Prof. Chu, Dr. Leong, and Michelle Chan. And I'll say thanks with that, and we'll now move on to our panel discussion sessions. So let me just stop sharing my screen here. And so just before we get started, if we could just have a quick introduction from both of our guests here, perhaps Olivia, if you'd like to jump in first.
Sure. Hi, everyone. It's really good to be here today. I am Olivia Ogilvie. I'm one of the co-founders at Oppo Bio, which is a New Zealand-based B2B supplier of cultivated meat cell lines. So very topical for the discussion today.
Great. And Giri?
Hi everyone, I'm Giri. I'm currently the senior scientist at Tree and have 10 years of research expertise in cell and molecular biology. And I have been contributing to Tree's cell-based dairy mission and have been working on different cell lines, culture media, and development of cost-effective animal origin-free culture media. And I'm very happy to jump on this discussion today.
Awesome, thanks both. So the first question, which I'll open to both of you, is around species selection. So given that one of you is coming from the cell line provider versus the cell line user for end products perspectives, how have you approached species selection for cell line development? And again, maybe I'll throw it to Olivia first here.
Yeah, definitely. So the species we work with are livestock at the moment. I'd just say that this is our initial focus. So we're working on pig, cow, and so ovine, porcine, and bovine. And with bovine, we're working with both Wagyu and Angus sows. We basically chose these species because we're a university spin out, and this is where we had been doing academic research and where our licensed technology was based. So we already had an advantage in these areas. Also coming from New Zealand, we have a pretty unique set of animals in these species. So we have ample access to all of these species, and since spinning out, we've actually formed partnerships with cow, sheep, and beef farms around New Zealand that allows us to have no issues getting access to these cells on an ongoing basis. I guess the third part of that is we surveyed the market and saw that most manufacturing companies, so companies who we're wanting to sell to as a B2B player, were wanting or growing, wanting to have research and products developed with these species. So taking all of those things together, we really locked in livestock, but in the future, we do hope to move into other species like fish and shellfish.
Awesome, and I guess Giri, how about your perspective as a user of these cell lines?
So as a user of cell lines, I would say, in my personal opinion, the species selection, it's all dependent upon the company objective and research focus and how they want to project themselves as a differentiated player in the market to provide specific kind of cell-based meat. So, and it all depends upon the accessibility to the cell line and how fast, what are the regulatory frameworks around the cell line and what kind of modifications have been done to the cell line. All these are to be taken into consideration before we select a species or a cell line.
Great, and I might just have one little follow-up there in terms of the live versus slaughtered animal question. I'd be curious about both how you see that and what made you decide to either go for live or slaughtered.
So live or slaughtered, I would say it's a concern based on the geographical location that you are staying with, because it's not necessary that you would have an access to the livestock at any point in time. For example, I'm staying in Singapore, so I don't have much access to livestock. So I would have to depend on saline providers for providing these salines. Whereas if you're in a geographical location, for example, if you are interested in some species that have specific geographical locations, then you have to depend on those saline providers for providing them the salines as well. When it comes to slaughtered versus livestock, it all depends on, I would say, factors like ethical considerations and the regulatory constraints around extracting these cell lines in from live animals versus from the slaughtered houses. And being in a location where I don't have access, I would have to depend on slaughtered houses for extracting those cell lines. Yes.
Yeah, so we have quite a unique perspective on this, having partnerships with farms throughout New Zealand. And we obviously have posed the question of live biopsies to our partner farms and had like big discussions around it. And at the moment, we isolate all of our tissue from slaughtered animals. And the main reason is that we've encountered some farmers, I guess they're not wanting to harm their animals when they're still growing up. To enter the food supply chain. So if we were to do a live biopsy, the farmer would have to take their animal to somewhere where they were able to do the operation, put it through that process, and then put it back on their farm to grow up and still go into the food supply chain. And that's because we're working with partner farms that are working farms. And so farmers have been really uncertain about wanting to do this. And so, yeah, that's why we're still doing slaughter biopsies at the moment. Yeah, it's definitely an interesting perspective and conversation to have with farmers about, can we just take that animal to a vet and take a sample from it? It'll be fine. And they're like, I don't want that.
So, sorry, just as a follow-up question, Olivia. So do you see that there being a concern or apprehension about the after effects of the biopsy, which is what farmers are concerned about? Or where does that apprehension stem from?
I think it stems from, yeah, the after effects. And often we might be taking the sample from a breeding animal, like a bull, and that animal's sole goal in its life is to provide that farmer with a lot of value and ongoing reproduction. And so it's a big risk for them to let us take the animal and put it through an operation and hope that it doesn't get an infection after. Yeah, and we did not expect that at all when we first had that discussion with the farmers.
Right. So do you see there, is that still an avenue that, you would like to consider, and you're working on it internally at OppoBio to sort of develop, say, better guidelines or criteria to inform them of how the welfare of the animal can be assessed after? Or do you think at this point you're sticking with slaughtered sources and that's it?
Yeah, it's definitely something we're working towards. I guess, yeah, we're working towards it and trying to talk to people more about it who have successfully done live biopsy isolation. So we haven't actually done a live biopsy within our academic research program or company. So just trying to get more feedback on what the process is like for the animal and what the actual risks are for the farmer. Because also our animals live outside in New Zealand. So if it's bad weather or rainy, they might have to be kept inside to keep the wound clean while it heals, which is not easy.
Right. All right. Just to sort of probe a little bit more into the research gaps that we're facing right now. it's a question for both of you. What sort of fundamental research gaps do you see as most urgent for us to address in terms of understanding how our cell lines function and what is required to develop high quality lines that are capable of, lasting the long run to get to commercially viable numbers and commercial production as needed? Have you identified any specific research questions that need that level of attention urgently?
Yeah, I can answer this first. So I think there is very little public and open source literature understanding of how these cells work. And that is a massive barrier to everyone's progress, in our opinion. So as a barrier, there's a massive opportunity here for companies to do amazing research and make some of those results available. I think a really good example of this is the RNA-seq paper by Messimer over at Mosameat. They did RNA-seq, and with the results, they were able to design an awesome media, cell culture serum-free media. And it's fundamental understanding of how the cells work on a metabolic level that I think can lead to discoveries that we have no idea about yet. Like, we can't even fathom what we could do if we could really understand how these cells work and grow on a metabolic level.
Do you see sort of metabolic modeling also informing other processes, such as on the media development side, for example?
Definitely. I think understanding the cells will enable us, will unlock our ability to grow them. And we know very little. So it's not surprising that scale up is difficult at the moment.
Yeah, I'm completely in agreement with Olivia. So understanding the cell line is really, really important, provided that we are developing cell lines from species which does not even have well-established characterizations set up, or a cell line stability that could last for a very long run. That's not there. So we would have to develop, there is knowledge gaps on how the cells function, understand the metabolic capability, and how each media optimization works could actually stabilize these metabolic changes within the cells. All these are gaps which we would have to understand. And the major gap is so far we have been using FBS for these cells to grow very well. But when we remove them, we see a lot of metabolic changes within the cells. How we can stabilize these cells in a long run without using any animal component is something to be more concerned about and to work on as well, to generate a database wherein the cell line and the characteristics are being well understood. And when it comes to a long run, there is a possibility of even doing immortalization works wherein these cells could be used for a very long time, and you need not have to bother on the animals for extractions of these cell lines.
Yeah, just as a follow-up question to sort of bring those two together, do you see the metabolic modeling approach also informing, for example, if gene editing were to become something that's more sort of adopted more in a more widespread manner, do you think that can actually inform what sort of targets a gene editing approach would be better suited to use for in order to improve performance? Do you see metabolic modeling also informing how, say, the lifespan or the health span of the cultures can be extended, perhaps?
Yes, I do see an interesting aspect to it. So when you go in for immortalization or do a genetic editing to this, you can build cell lines that can last for a very long time. That is certainly the major plus of doing a gene editing. But at the same time, what kind of gene editing can you do? Is there a regulatory approach to it? What will be the regulatory framework for it? On the pro side, definitely a gene editing will help you with understanding the metabolics of the cell to a great extent, and as well to overcome the current limitations that we have in terms of scale up, adaptation to an animal-free media, and cutting down the cost in terms of growth factors and all those stuffs. Definitely gene editing has a higher hand in the long run.
Yeah, I think we see that gene editing is a tool for the industry, and it should be used as a tool, but it doesn't guarantee success. So there's still a lot of challenges around gene editing. It's not a silver bullet. And we still need fundamental understanding around how the cells work to be able to produce kinetically stable, scalable cell lines.
Yeah, that makes sense. And do you think, or like, let me change up the question a bit. What do you think is the most optimal way for a cell line provider and a cell line user-based company to collaborate and to partner, not just on the testing out and validating cell line aspects, but also boosting some of these research gaps? Do you think there's space to collaborate in that manner? And what would be the best way?
Yeah, so I think this question is different whether you're a company or a researcher. So if you're a researcher, we have all of our cell lines available to purchase, all of our primary cells, sorry, not cell lines. So we have fibroblast satellite cells and pre-adipocytes from Wagyu, Angus, sheep, and pork, pig available to purchase, and you can purchase that and get them from us to do your research. So this is one way that we collaborate with the industry. If you're a company, this collaboration does look different. So as a company, your needs will probably be more variable. So the best way is to just start a conversation with a cell line provider. We're also a startup, and so we're trying to learn what the industry needs as the industry learns itself. And we're having early conversations with companies who want to work with B2B suppliers. And yeah, it's really just about starting those conversations so we can make sure we're designing lines that work for the industry.
Right.
So would you say, oh, sorry, just to follow up quickly on that, would you say at the moment that having off-the-shelf options for commercial customers is too early days in terms of it's not clear what the general requirements would be for such a product?
No, I think it's clear what the requirements are, generally at least. We do aim to have that by like late this year or early next year, but it's more our company is too early. We haven't validated the cell lines enough to feel comfortable giving them to a third party to trial.
Right.
Sorry, Giri, I interrupted you there.
No issues. So for me, like to collaborate with a cell line provider, the major objectives would be like what kind of species that they have. Do they have my species of interest? And then like what is the functionality and characteristics estimations that they have done through? Like have they looked at cell line stability? Have they looked at media optimization? Or is the cell line certified to be a halal or kosher? And some dossier around how the cell lines were extracted, from what tissue the cell lines were extracted, what tissue of the species the cell lines were extracted, and is there any IP restrictions around the cell lines? And so these are some of the things that we look for, and like how well the cell lines are growing, is it adherent or suspension, how it can adapt to our scale-up processes. This is how we look into when we look for a cell line provider or for specific cell lines with cell line provider. So these are some of the concerns that we normally put forward to the cell line providers as well, to get into touch with your cell lines, get access to that. Yeah.
Excellent, thanks. And to kind of change tack a little bit, as a question to you both, what gaps do you think governments or regulators specifically need to fill to help boost capabilities in cell line generation and banking, or making it clear what regulatory considerations these companies or researchers should be considering and working in this space?
So in my opinion, a regulatory constraints on like how the cell lines are being derived would be a good goal. I mean, good start to know how this align. I mean, what characteristics is the government and the regulatory bodies are looking for, number one. Number two is like a banking facility or a cell line provider facility within each geography will help ease the cell line, access to the cell lines. So that these two are major gaps that I'm seeing in terms of like cell line optimization and the scale-up process for cell line for the cultivated meat industry.
Yeah, definitely. I think there are obviously significant gaps in the information around regulatory and what tests are required for the cell lines to ensure their safety at the cell line generation and characterization process. We're working with a regulatory consultant to try and understand this. And I think it would be awesome if there was more publicly available information on the ingredients throughout the process. So not just cell lines, but how to regulate cell culture media, what type of tests need to be done there, how to regulate cell lines, rather than the process and end product approach that regulators are taking at the moment. But I mean, it's such early days in the regulatory landscape that it might be wishful thinking for now.
And I guess on that, do you feel that regulators are currently accessible enough in terms of, you know, being able to consult with them directly on these points, or is that another mechanism that would be useful?
I think they're accessible. They're more than willing to have conversations with companies, and yeah, they're pretty proactive about it in our experience.
Excellent.
So just as a follow-up question, Olivia, with regards to looking for regulatory consultants, do you find that there's a little bit of a handicap in terms of specific geographies that you're facing in understanding the regulatory considerations for specific countries or markets? Or is it do you feel like right now on the whole, it's quite on par in terms of like what the gaps are?
Yeah, I think when we are designing our regulatory strategy and making sure we're collecting the right data, we're doing it for Singapore and the US, just because that's where most of our customers and future customers are planning to launch their first products. So for us, it wasn't challenging to find people with the expertise and experience in novel foods in these two areas. Maybe if you were aiming for a more niche jurisdiction, but I can't really speak to that based on our experience.
Sure. Okay.
Great. Well, I think maybe we could jump over to the open Q&A session at this point. So I see there's already a bunch of questions in the Q&A box, so thank you for that. But as we go, please keep adding them in, and also for everyone else, you can vote up questions you do want to see answered, as we're definitely not going to be able to get to everything. So just a really quick one. Nicholas asked about whether there's a point of contact at GFI to ask about regulatory requirements. That is one where I mentioned we do have a database around regulatory consultants. So that's not publicly available, but if this is a point you'd like to learn more about, you can contact any of our CM or cultivated meat SciTech specialists, and we can share that with you as an initial point. And we are also open to having conversations internally if there's other elements that might be useful from us. Let's see, the next one here we have is from Nikita. Do you foresee the opportunity for such B2B players to be startups or incumbent cell line providers to expand their current product offerings? How can startups compete with existing players in this regard? So maybe Olivia, as a startup in this space, I might open this up to you first, and then Giri, if you have any thoughts on this as well.
Yeah, for sure. I think this is, for us, it's like as a company, it's a definite risk, and we think about it a lot. So we say, as a company, our biggest competitor is the existing cell line development programs within companies. Maybe the reason that we maybe don't see this as a massive risk is we see a lot of these companies approaching us and telling us that they have problems with their cell lines that they can't solve. And so I think some companies will pivot, especially as they start to feel more commercial pressures from their investors to make some money. But a lot of the companies have invested heavily into end product manufacturing, and pivoting just isn't in their intellectual property protection strategy. They're trying to hold everything as trade secrets, and lots of those companies will do that until they succeed or fail, I think.
Yeah, Giri, I'm not sure if you've had to interact with both startup or large potential cell line providers, and maybe you've had variable experience with both you could speak to.
So it depends upon, like, in the sense, like what kind of cell lines we are trying to procure, or like it all depends on what kind of cell lines and from what cell line providers are willing to provide us. All matters actually. And I would say, in if you talk about like genetic engineering approaches and all those, it not necessarily be genetic engineering approaches, but also like immortalization activities that have been done by cell lines could be all immortalization by spontaneous ways could also be helpful in developing cell lines that could be of long-term usage for different companies. It not necessarily be GM approaches as well. And the optimization of cell lines differ between different companies, and like definitely, as Olivia said, not everything is being openly collaborated between the parties, which open collaboration would definitely promote a more successful environment for the cultivated meat industries.
And I guess just finally, from the GFI perspective, we have discussed cell lines as being a bottleneck for the sector for years at this point, and we haven't seen a lot of movement until very recently. So I think just as a general indication, we've seen that incumbent players haven't moved into this space for whatever reason, where we've seen a lot more movement when it comes to culture media developments or bioprocessing developments. Lately, we have seen a few more startups and companies get involved in this space. So it seems like maybe we are starting to see some movement, but it's more from that startup end of the spectrum.
Yeah, sorry, just to add on to that, in terms of existing cell line providers who cater to like the biomedical science research field, for example, from my sort of limited conversations, I get a sense that there's a bit of an apprehension to enter into a new space that from their business point of view, they're like, well, we don't really need it, you know, so they don't, the real need hasn't, I guess, hit them. And the fact that there's actually a huge white space with a lot of revenue to be gained hasn't really reached them yet, perhaps. But in a way, I'd also like to look at that as a silver lining for startups looking to enter this space, because that's a huge opportunity for them. So it is a little bit of a win-win depending on who you speak to. But I think it's been really encouraging to see, for example, Extracellular, Dragon Bio, a couple of new players who've pivoted their work to now cater to this white space. So I think the next few months and the year to come, it'll be interesting to see how those offerings perform, and for companies that validate those offerings, what their feedback is in terms of what else is needed. So I think it's a good movement in the positive direction.
I see there's a few questions around obtaining cells from live or slaughtered animals. There's also ones looking at the age of the animal. So I guess as a general group up of those questions, do you see different performance if you're sourcing cells from live or slaughtered animals, or perhaps young versus old animals, or different biopsy areas? Obviously, you know, if you're sampling muscle versus fat, you get different cell types, but maybe different areas within the same muscle tissue, does that show variability?
Yeah, so we haven't done the live versus dead, obviously, but we definitely see significant differences between individual animals. As far as I'm aware, there's no published literature on this at the moment that shows how big those differences are, but it's not surprising that there's variation within populations, even populations like a population of cows that's a closed herd, for example, you see individual variation. We don't really know at the moment how to predict that, or what the trends are, I would say. But like, I think there's a lot of opportunity there for cell line providers, companies like us, to try and work that out and find the best animals, the best breeds, the best tissues, the best ages to get the best cells from. And maybe this is what Mosa Meat is doing. If anyone is on the, from Mosa's on the call.
Yeah, I think anecdotally, I've heard similar from some companies where they'll do repeated biopsies of the same type of muscle tissue, but they will get variability in the actual efficacy of those different cells that they're getting out. And so just through random repeated biopsies, they'll select whichever comes out with the best performance, but not truly understanding why that's occurring. So again, yeah, I agree. That's an area where I think more understanding of individual cell differences would be beneficial.
Yeah. I think there was a preprint that was released maybe 2 years ago that had 3 biological replicates in. It didn't get through to full publication. It was from an employee at Mosa who was doing a project in it. I can't remember the title, but I can have a look and send it out if anyone's interested.
And just looking through it, more questions here. If you've got one, also feel free to jump in.
Sure, I'm busy typing in answers for some of them. There's a question from Satish that says, does GFI help startups in organizing seed capital and venture funding? I know this is out of syllabus question. No, that's fine. That's a good question. So we do have a corporate engagement team that can help answer some of these questions. So we don't directly help with the capital, seed capital or funding itself, but what we can help is link you to some VC firms and grant calls which are ongoing, and even maybe help review some of your pitch decks, etc. So we can help provide some support. So if you're keen to engage with our corporate engagement team, just get in touch and we'd be happy to take that conversation forward.
Yeah, and maybe a cell line question here from Sid Jane on a similar line. Is it fair to say that startups or the work being done is more around R&D and early stage R, meaning it is hard to attract investors in cell line development companies? And how can we attract them? So I guess, Olivia, how have you found attracting investors around your work so far?
Definitely. I think so. We are really lucky in New Zealand that there's a lot of investors who are really interested in supporting technology companies. We have a lot of food innovation here in New Zealand, and people understand how hard it is to commercialize technology and build a large food marketing brand. So our approach of being a technology enabler was taken quite well. I'm not sure if it's the same in all jurisdictions, but I think there's definitely an awareness of the need for enabling technologies to scale the industry, and people are wanting to fund that. Yeah, it's about having the technology working enough. And I think reports like the one that GFI is releasing will only support enabling technology players. Like your guys' media report and the cell line report will help give investors confidence that this is a commercial opportunity as well as a technical opportunity to make progress.
Awesome. Yeah, I think generally, maybe not specifically looking at cell lines, but the general conversation around investment, we have seen more of a focus around picks and shovels, as they call it, which is the kind of enabling technologies as opposed to the end product producers themselves, which is great to see that kind of diversifying conversation when we look at investment in this space.
I'm just going to club a few questions I see on the topic of halal here. For example, there's a question that says, can we use plant-based FBS in culture media? And how does that relate to halal, I think? And then there's another question that says, if a cell source is an embryo, would it come under halal or kosher? So there is no clear regulation or guidelines to sort of determine what qualifies and doesn't qualify as halal with regards to the cultivated meat production process yet. So these are questions that we also have and are trying to understand how best to apply the concepts of these certifications to the scientific process. And we do have ongoing conversations with different organizations to get more information on this. So I'm afraid we can't give you a fixed answer right now, but I would say keep an eye out in the coming months. As and when there is any information, the GFI team will share on all of our platforms.
And I guess something I can share is that we are looking to have a webinar in the next month or so, specifically on the topic of halal certification and cultivated meat with a relevant religious expert that's been looking into this. So if you do want to learn more, just keep an eye out on our media channels for that.
There's also a question from Jamal here. Hey, Jamal, nice to see you. How has chromatography and mass spec been used for metabolomics for this up until now? Olivia, Giri, do you both have any insights into this?
So
I think you go first, Gary.
Sure. To speak on the metabolomics part, we so normally on the media, when you're developing the media, you could look at the different metabolites that is being released by these cells and look for optimization conditions and see how you can support these cells for long-term stability. And that's where, and when it comes to mass spectrophotometry, or like using mass spectrophotometry, like you could look for like what kind of proteins are being produced, what kind of fatty acids being produced by these cells, and how you could enhance these fatty acid contents in these cells to or protein contents in these cells as compared to the tissues that are, or the meat that is normally available on shelf, and try to match it by means of media optimization process. So that's how the higher techniques could help you in meeting the nutritional requirements and enhancing the product quality.
Yeah, I guess very similar. We use techniques like LCMS and GCMS to look at the flavor and nutritional profiles within our cell lines. And it's super exciting because my PhD was actually in this space, but looking at proteins. So I get to do a little bit of science when we do that work, which is cool.
Awesome. I see there's a question here from Calissa Lim asking what topics should be explored further, especially in regard to regulatory coordination between jurisdictions regarding cell line requirements. So on that, in the presentation, we mentioned a few areas where companies specifically said they had the least understanding of requirements around documentation for isolation. Sorry, no, that's one where they had the information around the techniques that can be used for cell line generation, etc. But in terms of coordination between jurisdictions, something that we've been seeing is more governments are beginning to look seriously into setting up their regulatory regimes for cultivated meats. So in the APAC region, Japan and Korea have really pushed ahead. Australia is assessing its first product. Malaysia is just starting to look at what it could be doing in this space, and Thailand as well. So I think having coordination, not necessarily of making sure one jurisdiction's approval will relate to another, but at least having these regulators discuss what type of requirements they're looking to put out, and seeing if they can align them where possible, or sharing information with different jurisdictions that might not be so far down the track of reviewing the literature, understanding the challenges and what specifically should be required from a hazard analysis, food safety perspective. And so for a jurisdiction like Singapore, where they're a leader in this space, we really encourage them to have these conversations and share with different regulators now, when they're just beginning to explore this process and set up their regimes. And different groups such as us and the APAC Society for Solar Agriculture do want to support this, and companies do want to help push this conversation forward. So I'd also suggest that having more regulator, private sector collaboration on this topic would be really important as well. But I'm not sure if anyone else wants to jump in on that one as well.
It would be awesome to have some sort of guide like they have in biomanufacturing for cell lines, and the types of tests and standards that we need to use to test them. And I'm hopeful that will happen eventually. It's happened in biologicals, and so I expect it will happen in this area when we have some more case studies on the food safety risks in different lines and how to control them.
I just saw there was an interesting comment in the chat section from Vin. I think one common skepticism for cell line providing companies is that there isn't a clear business model. I've heard pricing for private companies anywhere from a few $1,000s to a few hundreds of thousands of dollars. So if pricing is sorted out to be more reasonable, then there's likely to be a market for cell line providers. So yeah, Olivia, I'm curious to hear your thoughts on this and how you've sort of worked towards sort of bridging that pricing issue.
Yeah, so we think about the pricing in two different ways. The first way is for research use only. So for researchers, we have one set of pricing. So that's very similar to if you were to buy cells from ATCC, around $700 US per vial of cells is what we're currently selling them for. Having the cells for commercialization is a very different pricing model. And I guess it depends how much work has gone into developing that cell line and what we believe the value of that cell line is. If it's super high performing, it's more valuable, and it will hopefully help you more. And so you should be willing to pay a lot more for that. I think the pricing is, you should think about it if you're a company. If you have a whole team of people and set up a lab to specifically do cell line development, or you could pay something like $60,000 a year to outsource that. I don't know, would you? Because I definitely would. And when people talk about the cell line development industry and cultivated meat not being big enough, we always point to the comparable industry of cell line development in the biomedical industry. I think today it's about $5 billion, with $5 billion, and cultivated meat is set to grow to be a bigger part of the manufacturing industry, hopefully, than biologicals in the future.
Excellent. I think we've just run out of time there. So I'd like to thank Olivia and Giri for joining us, and everyone in the chat here for watching the recording and asking some great questions. As we've just posted in the chat, the recording of this will be available on our YouTube channel shortly. So please review that if you want to, or share it with others that might find this interesting. And please have a look at our report as well if you'd like to see some more detail on different topics that we didn't necessarily cover today. And with that, I'll just say thank you to everyone. If we have questions that we didn't get to, feel free to reach out to us at our emails that have been shared in the chat as well, and we could discuss that in a follow-up conversation. Thanks, everyone.
Thank you, everyone. Thank you, Giri. Thank you, Olivia.
Thank
See you.
Bye.
Bye.